thank you for joining. It is a lovely day here in London, so I do appreciate you guys joining in. I'm Mariam. I'm leading the hackathon series today. I'm the co-founder and director of Enteromics, which is a medtech startup that develops smart gut sensing pills. But alongside my role at Enteromics, I also research biocrime and the Internet of Medical Things and cyber biosecurity as part of my PhD studies, which really focuses on aiming to inform evidence-based policies regarding biocrime. And this includes the Ingestible Things, or the Internet of Ingestible Things hackathon, which is a series of workshops that brings together experts from cybersecurity with the medical device industry and makers to really help design security for the ingestible things, which are internet-connected, smart gut sensing pills or ingestible medical devices. So please do register for the hackathon that will take place in May. So I've just posted a link for the registration. And if you are interested, I have just posted another link on more information on the hackathon. The winning team does get a cash prize of £1,000, sponsored by the Daw Center for Future Crime here at UCL. The aim of the center is to focus on key questions like which emerging crimes should we focus on given limited resources, or how do we mitigate future threats? Right, so as a prelude to the hackathon, we have a series of talks which really explore the prospects and the challenges of the ingestible things. I am pasting as well in the chat the rest of the talks in case you'd like to sign up. Today, the structure of the webinar will include a 20-minute talk from our guest speaker, followed by a 15-minute Q&A. So please do write your questions in the Q&A box within Zoom, and think about the takeaways in the end. Right, so second in our series is Geoff Briggs. Geoff spent almost 30 years conducting scientific research in an academic environment, developing his skills as a molecular biologist and biotechnologist, with 13 of those years dedicated to the study of bacterial DNA replication and repair. In 2014, he left the academic world to join Walgreens Boots Alliance, and has spent 7 years discovering, evaluating, and developing new technologies in consumer skincare, really focusing on beauty devices, consumer diagnostic services, and the skin microbiome. As part of the skincare innovation team, he helps people transform their skin health and skin longevity in ways they never thought possible, drawing upon the knowledge he gained during his academic career with his new insight into the commercial world. So without further ado, over to you, Geoff.
Thank you. So I said, I'm not an expert on the gut microbiome, though that is what we're talking about today. I have done a lot of reading, but everything I'll be presenting today is other people's work. I'd just like to hasten, none of this is done by myself. It is other people's work, but it's an understanding that I've built up as part of trying to understand the human microbiome as a whole. So if I go to our first slide. So hopefully that's got the right screen on everybody's screen. I'm sharing the right one.
Yeah,
so
perfect.
Why should the public want to know about their gut microbiome? Why do the public want to know about their gut microbiome? That's because it is all important, and it tells you far more about you than pretty much anything else, and that includes genetics. And as a molecular biologist who's spent a lot of time working in genetics, that's actually fairly hard for me to accept, but microbiome is more important than genetics in most cases. So a few numbers. We are not who we think we are. What we are is an ecosystem, is a community. In fact, it's not just an ecosystem, it is a collection of ecosystems. We are, of our cells, we have more bacterial cells than we do human cells within our body. Over 90% of our DNA sequences, so that obviously provides our capability, our metabolic capability. Over 90% of those that we have in our body are bacterial, they're not human. Then you add in the phage, archaea, yeast, fungi, mites, all those other bits and pieces that call our body home. Phage is the unknown space really here, and we are yet work out how many phage species there and phage DNA there is that are part of us, and that is the newest area of research. Vast majority of these bacteria that call us home live in the gastrointestinal tract, and specifically the lower intestine, so the colon. And microbiome is one of those things that everybody will know about by now, but it is this ecosystem. It's an ecosystem of microbial species which exist in a specific environment, and it involves that environment, and it involves how they interact with their host habitat. So it is not just the bacterial species themselves, it is how they fit in with a local environment. And we are a collection of ecosystems. So the best known are the ones here. So oral microbiome, placental microbiome, gut microbiome, skin microbiome, vaginal microbiome. But to date, every part of the body has been identified as having a microbiome. The cancer tumors have a specific microbiome which is specific to cancer tumors. I think the best one I like is the brain microbiome. You have bacteria living in your brain, and they don't just live there. What they do is they affect how you think. So you are even thinking bacteria. You're not just thinking human, you are thinking bacteria as well. And a very quick lesson on taxonomy. So when we're talking about bacteria, we have the standard things. We have domain, kingdom, phylum, class, etc. And just to put these into context, on the left-hand side, we have the taxonomy of dogs. And so you start off with eukaryotes, and then you have the animals, then the ones with backbones, then you're down into mammals, carnivores, canines, and all the way through. And bacteria work in the same way. And a lot of work that's been done in this space, or a lot of people, when they talk about this, they talk at a very high level. They're talking at a phylum level. For example, we've got firmicutes here, and that is the same level of differentiation as creatures that have got backbone and not got a backbone. And we are talking at that level of differentiation. When we get further on, some of the talk I'll be talking about is sequencing and how we talk about sequencing. And at best, most of that is at the genus level. And that is down at as far as the dogs are concerned, that is all dog species. So that is dogs, that's wolves, that's coyotes, that's jackals. These are all very, very different things. And they behave differently. And just to put that into even more context, not all species or strains are equivalent. When you're talking about probiotics, they tend to look at the side and say it's lactobacillus, and that is at a genus level. You need to be looking at species level, you need to be looking at strain level. What we've got here is Danny DeVito and Kevin Garnett. It's a photograph that Danny DeVito took where he ran into Kevin Garnett at an airport, I think it was. And these are both Homo sapiens, subspecies sapiens. They are both at a strain level, and they're very, very different. You know, if you're looking at these and you're saying, who do I want on my basketball team? You would not go for Danny DeVito. So when you're looking at what species, what strain should I be having to have a function in my body, or what species has or strain has a function in my body, you really do need to be looking at that strain level. When you're looking at purely genus level, it is not enough to tell you what something is going to do. So we're here primarily talking about the gastrointestinal microbiome. So from gum to bum, this is not my phrase, this is something I've stolen from somebody else's. But here, when you're talking gastrointestinal, you're talking all the way through from the oral cavity, the esophagus, the stomach, and then small intestine and large intestine. It's important to remember that your gut microbiome is not just the colon. It is far more than that. And when you're talking about anything to do with your gastrointestinal tract, you're talking about all of these. Now, 95, 98% of these bacteria are in your large intestine, but it's worth remembering these other parts as well. And again, we'll discuss that a bit later. And if you look at the gut in particular, you do have your small intestine, you have your large intestine. Your small intestine has a different environment to your large intestine, which has a different environment to your rectum. And the bacterial species that live there are different as well. And not only that, there's another level of differentiation. When you get into your colon, you have your mucal layer. So those are the bits which are part of the epithelial, and you have those which are in the digestals. So that is the sort of the main flow through of food and digestive material that lives in the digester. And these are different environments. And this again is going to become very, very important later. So why am I, why WBA interested in the human microbiome? Well, most of the stuff that we do is looking at taking people from an unhealthy body state, whether that is skin, whether that is gut health, whether that is vaginal, any of these things. We're taking us from an unhealthy position to a healthy position. That's what we're aiming for. And primarily, in the past, what we've been doing is treating the body itself. And so you've been going from that bottom route, so that bottom right route. So you're going from the unhealthy to healthy as far as the body is. So what discussion the microbiome does is it gives you another route through, and something else which you can use to move people from a healthy to from an unhealthy to a healthy state. And that's by looking at the microbiome as well as the human part of the body. We have a problem with this, especially when you're talking about the gut, but it goes for the skin as well. We don't know what healthy is. And what is a healthy gut microbiome? We don't know. We probably know what an unhealthy microbiome is, but we don't know what a healthy microbiome is. What we've got here is some work that was done a few years ago. And this is looking at the bacteroides and firmicute ratio. So this is, we have to look at the little chart on the right-hand side. That is at the phylum level. If you remember back to the taxonomy, that is the difference between backboned creatures and non-backbone creatures. We're looking at that level of differentiation. What we have here are 98 healthy volunteers aged between 19 and 39. And you can see what we've got here is a ratio of your firmicutes, which are the green, to the bacteroides. And it goes from almost 10% one, 90% the other, all the way up to almost 99%, 1% other. All of these people are considered healthy. And this is at the phylum level of differentiation between them. So how can you say what is healthy? Another problem, it changes as you age. Your microbiome will change as it ages. And so if you have a young gut microbiome in an old person or an old gut microbiome in a young person, they may be wrong, even though it could be what you had or will have at that stage. And so you've got to remember that as you get older, your microbiome becomes more and more specific to you. What it is those bacteria have been living and evolving in your gut for 20 years, 50 years, 80 years, whatever. They will be specific to you. They will have gone through genetic mutation and modification and evolution, and what you have is specific to you and different to everybody else in the world. And so what is healthy for you is what you've got, not is or likely to be what you've got, not what everybody else has. Second problem we have when discussing the gut microbiome is the general hypothesis you have is healthy microbiome means a healthy gut, which means healthy body. In the same way as healthy gut means healthy body, means healthy microbiome, and so on. But this is the huge problem we have with all of microbiome work. Correlation does not mean causation. We can link a condition of the microbiome, so a microbiome state, with either a healthy or an unhealthy body function, whether it be skin, whether it be gut, whether it be oral, whether it be dandruff, whether it be vaginal. We can say, oh, look, they have a healthy body, the healthy gut, and these are the microbiome that they have. They can say, oh, look, they're unhealthy gut, and these are the microbiome, and you'll see a difference, but you do not know what comes first. You do not know whether your healthy microbiome is because you have a healthy gut, or your healthy gut is because you have a healthy microbiome. And this is because you're looking at environment. The environment of a healthy gut is different to the environment of an unhealthy gut. Therefore, the bacteria that live there will be different. Remember, we're talking ecosystems. And different ecosystems will have different creatures that can live in them. And it's the same as far as your gut microbiome is concerned. So why are we interested? Well, why are we interested in the gut in particular? The gut, obviously, there's a lot of gut conditions, guts and gut health is important, but what happens in the gut does not stay in the gut. And that, again, is a line that I've stolen from somebody else. That's not mine. But there's been good evidence that the gut microbiome has effect elsewhere in the body. So obviously you have the gut health, so IBS, IBD, Crohn's, celiac, food intolerances, but it's been associated, correlated with asthma and allergic rhinitis, diabetes, obesity, acne, eczema, cancer, lupus, autoimmune disease, stress, sleep, diarrhea, constipation, kidney disease, liver disease, all of these things, and also how susceptible you are to other infections like norovirus, like influenza, even like COVID, which is obviously the current one. These all depend on your gut health and what's living in your gut. And there is the gut-skin axis, there is the gut-brain axis, there is a gut-skin-brain axis. Probiotics that you take orally can change your vaginal microbiome. There's no direct link, but they will change your vaginal microbiome. And so there are a number of ways that what happens in your gut does not stay in your gut. It interacts with your whole body. It has an effect on your whole body. How does it do this? Well, primarily, it is to do with short-chain fatty acids. So these are butyrate, propionate, acetate. These are the products of fermentation of fiber. So that fiber you're always being told to eat is not for you, it is for your bacteria to eat. And what they do is they convert it into butyrate, propionate, acetate, and other short-chain fatty acids. And these are the signals that have an effect on the rest of your body. So butyrate is probably the most important. This is involved in signaling pathways. It is also heavily involved in gut health, and because this is the food source that your epithelials, so those cells in your gut, use to keep for their standard metabolism. But short-chain fatty acids don't only stay in your gut, they do enter your bloodstream, they do go elsewhere. It's not as simple as pure butyrate. It's not as simple as these bacteria make butyrate, therefore it's good. Because butyrate is just part of a huge network. Remember, I talked about this being an ecosystem. It is an ecosystem, and things talk to each other and they interact with each other. And so, yes, we can have butyrate producing bacteria in your gut, and that's great, but they don't necessarily make butyrate directly. They need other bacteria to metabolize your foodstuffs into something which they can then metabolize to produce the butyrate. And we've just got a very rough network chart here. And you have your acetate producers, you have your acetate utilizers, you have your propionate producers and your utilizers. Lactate is there, succinate is there. All of these things form a network. And so just having individual species, when you talk about individual species in probiotics, probably doesn't work. What you need is to consider this as a whole, as an ecosystem. For example, Lactobacillus, which is the common gut microbiome probiotic, doesn't actually produce butyrate at all. But what it does do is it produces the lactate, and the lactate then feeds into the rest of the system. So I talked about short-chain fatty acids, I've talked about lactate, and I've talked about butyrate, and I've talked about propionate. But there are another key set of signaling molecules that are used. So the other key one here is bile acids, and there is a an intrinsic relationship between bile acids and the gut microbiota. It is a close relationship, it's complementary. Bile acids control gut microbiome overgrowth and they have an effect on your gut health. But your gut microbiome is involved in the metabolism of bile acids and changes what the bile acids are converted into. And bile metabolism is key to lipid and glucose homeostasis, which are then talking about the entire body. You're talking about obesity, you're talking about diabetes, talking about all of those bits and pieces here. So bile acid, the relationship between bile acids and the gut microbiome is very, very important too. And then we have choline. Choline is essential. It's there as a brain signaling molecule as the acetylcholine, but there is a problem with it. Yes, it's involved in lipid metabolism. It's important for that. It's the neurotransmitter, one of those things we get from red meat. But the gut microbiome, what it does, it converts it to TMA, trimethylamine, and that is then converted in the liver to TMAO. And there is clear correlation between increased levels of TMAO and incidence and severity of cardiovascular disease. And this is probably, the Western disease problem that we have is on the Western diet. This is why we have excess of heart attacks and so on. And this is how that comes apart. This is the problem with the Western diet, is the TMAO that gets into your bloodstream, and it only gets into there because of the way that it's metabolized by the bacteria in your gut. So we've talked about these things. There are a couple here that we've mentioned. We talk about immunity. And it's oft mentioned that 70% of your immune system resides in your gut. And this is primarily true. And the microbiome interacts with this both directly, because your immune system has little sensors that go out through your epithelial into the gut tract, and they identify what's in there, and that is how they get the antigenic sampling. But in addition to that, if you remember, I said that butyrate, that short chain fatty acids that's produced by your gut microbiome, that is the thing that keeps your epithelial cells healthy. And if you haven't got healthy epithelial cells, then what you do is you have leaky gut syndrome, and the wrong things get through into the system. And that butyrate is also the energy source for a lot of your immune system cells. So without butyrate, which you only get from your gut bacteria, then your whole immune system starts to fall apart. And there's been good links to show that, for example, probiotics link to upper respiratory tract infections. There's the link with COVID, which has been talked about. All of these things are here because your immune system is based in your gut, and the health of your gut is based on the microbiome that lives in your gut. And it does go wrong. So IBD, Crohn's disease, celiac disease are all autoimmune diseases, which celiac is the best understood. Note they are different to IBS, which is irritable bowel syndrome, which tends to be a bloating effect, and that's due to incorrect fermentation. Whereas what we're talking about here is much more serious, and this is an immune disease. And celiac is a paradigm for that. It's the one we understand best. It is likely that what happens is you have poor, unhealthy epithelial cells, and that allows, because of your gut health and the fact that not producing those butyrates to keep it healthy. With unhealthy epithelials, you get leaky gut syndrome, and those gluten molecules get through from your diet, through into your bloodstream, through into the immune cells, and they trigger immune response. And that is where you get your immune response. That's where you get your inflammatory bowel disorder. You get problems like celiac. And obviously, we talk about this as a gut microbiome, but the mechanisms here are also relevant to other autoimmune diseases: lupus, ALS, arthritis, osteoporosis. All of these things are autoimmune diseases and likely to be caused by leaky gut and your immune system based in your gut being incorrectly primed because of antigens that are getting through the system which shouldn't get through. So we mentioned diabetes a couple of times, mentioned obesity. The gut microbiome modulates insulin sensitivity in numerous ways, both upregulating and downregulating. So this little table here, we've got short chain fatty acids which are on there, and they have a positive benefit. Vitamins, which are, a lot of the vitamins are produced in the gut, they have a positive benefit. But we've just talked about leaky gut syndrome, and that has a negative effect on insulin sensitivity. The amount of carbohydrate that gets into your system is so associated with insulin sensitivity. We mentioned previously about bile acids, and bile acids regulating your glucose levels, your lipid levels, and bile acid and the microbiome are all, intrinsically associated with each other. So when you talk about the link between gut microbiome and obesity and diabetes, it's there, multiple pathways, but it's there, and this is why the gut microbiome is so important. We have a obesity problem is in the Western world again, which is huge and causes probably more deaths than anything else, and it's all due to your gut. So we've talked a lot about what happens in the colon so far. Just a couple of things to point out. The colon is not everything. So lactose intolerance is obviously very, very different to gluten intolerance. Lactose intolerance is actually a small intestine problem. And what happens is ordinarily lactose, it's converted to glucose and galactose in your small intestine, absorbed into your body, and everything is wonderful. Lactose intolerance is because you have problems in your small intestine, which means that lactose is not metabolized properly in your small intestine. It is not taken into the bloodstream, and it gets through into your large intestine, and the bacteria there have this food source which they're not used to, they're not supposed to have, and they metabolize it, and part of that produces gases, produces methane, it produces carbon dioxide, it produces all these things in your gut, which gives you your, it causes irritation and bloating because you've got a problem in your small intestine. This is actually, we have another one here, SIBO, small intestine bacterial overgrowth. And this is another problem that's out there, and this is a small intestine problem. And this is primarily, it's not due to any specific bacteria. What happens is that the bacteria that should live in your large intestine migrate and colonize your small intestine. So there is no clear cause for this other than what would be healthy in your large intestine and you need in your large intestine gets up into your small intestine, and it interacts in a different way with everything else. It interferes with your small intestine function. So again, not just the colon. Small intestine is important here as well. And that comes into what I'm about to say next. So I hope I've told you why the public should want to know about the gut microbiome, how important it is. The little quotation from a paper up there, microbiome in your gut could predict whether you're likely to live for the next 15 years. And the more we understand about the gut microbiome, we understand how important it is and what it can do. So if we understand how important it is, then surely the consumers want to know, okay, tell me about mine. Unfortunately, the technology currently is limited in this space. If you do Google searches, even if you look on Amazon, various things, there are literally hundreds of companies which will offer you as a consumer, as a member of the public, will offer to analyze your gut microbiome and tell you what's in your gut and draw conclusions from that. And some of them are very good quality companies, and their science is excellent, but they tend to use a 16S ribosomal sequencing of fecal matter. A large number of companies offering this, as I say. Now, there are problems with 16S ribosomal sequencing, which are common and well understood across all areas that do 16S RNA sequencing as far as microbiomes are concerned. It tends to resolve to genus level only. So there's limited species resolution and no strain resolution. And if you remember, referring back to what I said earlier, you need to know at least the species, because let alone strain, you need to know at least the species. Genus is not accurate enough to tell you what these bacteria are going to do. 16S only identifies bacteria. It does not tell you about your fungal composition, your archaeal fungi composition, your phage composition. It doesn't tell you about any of these others. There are additional methods to analyze and tell you about these, but that becomes more expensive and they're not offered routinely and will not be at a cost that the consumer is willing to accept. It's not a quantitative method. Results are presented as a percentage. So you'll say you've got 80% of this, 20% of that. But that doesn't actually tell you how many you've got there. And if you lose a species completely, obviously the percentage of the other one goes up, even though the number of the other one may stay the same. So it is not quantitative, it's qualitative only. And so it's difficult to really draw conclusions from this. You're looking at 16S sequencing, you're looking at sequencing the 16S ribosome, this region of the bacterial DNA, and looking at searching against the database, which means it can only tell you about the bacteria that are already in the database. Otherwise, which means if your bacteria that you're sequencing is not in the database, it can't tell you that it's there because it can't recognize the sequence. It's looking at DNA only. Doesn't differentiate between what bacteria are active, what bacteria are viable, i.e. alive but but maybe stationary in a stationary phase, or dead and so having no function and not possible to have a function, or even live cells. All of these will have DNA in there and can be recognized, and you can't differentiate between these. And this comes, there's a case here, Lactococcus lactis. It can 16S sequencing will pick up the DNA from your food. And some early studies were noticing abnormal results for Lactococcus lactis in there as a bacteria. And they couldn't work out why. And then they pulled it down to the fact that these people had a high milk diet. And so the bacteria from the milk, the DNA from the bacteria in the milk was getting through and being read at the bottom end of the system through 16S sequencing. And this obviously goes for any of the foods any foodstuffs you eat. The bacteria, the DNA in there could theoretically come through your system, be picked up on this, analyzed, and give you an incorrect reading. So that is where we are. That's problems with 16S. We then have problems which are specific to your gut. So we have the limitation of 16S ribosome sequencing, but the big problem is all these deal with fecal matter, i.e. the stuff that falls out to your bottom, to be quite honest. It is the bottom of the intestinal tract. And this gives a bias. It absorbs the longitudinal variation and the abundance of bacteria species along the length and width of the gut. I hope I've made it clear that your small intestine is important as well. It measures the stuff in the digestal region, so that the bit that comes out the bottom, rather than the mucosal surfaces, the bits that are in the epithelial folds. And they are perhaps the more important ones. There's a recent paper that's been published which proved that the rectal mucosal microbiome, but not the fecal microbiome, could be used to detect colitis. And the stuff that comes out the bottom, but when, as a consumer, you get your bit of paper and you fish around in the toilet bowl and you take your sample, none of which sounds very nice and is another problem with this sequencing. the method of identifying. But what you're pulling out there is not your mucosal layer. It is not your small intestine. It really is saying only what is at the bottom end of your gastrointestinal tract and in that digestible region of it. And perhaps most important, it doesn't measure metabolism. You know, all of these things, the bacteria are important, but they're not everything. It is their metabolic activity, it's their metabolic potential, it's how they interact with each other that's important. The bacteria themselves that are in your gut do not go up to your brain and change how you think. They do not go to your skin and affect how you respond to acne and eczema. They do not go to your liver and cause problems. If they do, then what you've got is sepsis, and that's bad. Bacteria in your gut, the actual bacteria should stay in the gut. How they talk to these other organs is through the metabolites. And just gut microbiome analysis will not tell you what metabolites are there. They will not, you can predict what could be produced, but they will not tell you what is there. They will not tell you what is being synthesized. They will not tell you what is entering the bloodstream. So there are some alternative technologies that are out there. So people are working on blood analysis, and actually from the blood metabolome, you can predict to some extent what the gut microbiome is by looking at the metabolites that enter the bloodstream. There is Food Marble, which is a company which offers a gas sensor to monitor gut microbiome. So you breathe into this sensor and it tells you from the gases that come out, that it tells you or can tell you something about the health of your gut microbiome. And the breath test is common for SIBO, that small intestinal bacterial overgrowth, because it can recognize there are gases being produced in your system which shouldn't be there, because the bacteria are higher up in your system and are interacting and producing gases that you shouldn't normally have. Other companies, for example, Milico, which is spin out of Imperial College, is working on urine testing and looking for metabolites in the urine test. And they do this via an NMR machine. And from this, they can identify what you really eat. And obviously, if they're identifying what you really eat, part of that is how it's digested by your gut microbiome. And so this is another technology. So those are alternative technologies. That's not necessarily what we're here to today. So what is the future in this space? So what we're looking at, and Enteromics are involved because this is the space they play in, is an ingestible sensor, an ingestible way of measuring. And there are lots of companies out there are playing with it, playing in this space. And they could be sampling the gut microbiome itself. I've seen some of these little pills that you take, and they have little almost pockets that open up depending on the pH, the area that you're in, or on a time basis, and they take a little sample of the gut microbiome as it goes through your system. And this way you can get samples from the small intestine, you can get samples as you go through your system. There are others which will measure pH, there are others which will measure the bile acid metabolism, all these sorts of things which give you some idea of what is happening through your gut rather than just what's coming out the bottom end. And these are important. This is the way the way forward. One of the big problems with here is, or a lot of them, you end up needing to collect the sample at the end, and that perhaps is even more horrible than scraping a sample off a bit of paper and putting it in a tube and sending it away. So there's still place to work in this space, but this is potentially the future of gut microbiome analysis, and hopefully that's what we'll get out from today. I think that's my last slide. Yeah.
Perfect. Thank you, Geoff. Thank you. That was quite, you did a really good job communicating a lot of complex science there. So I'll open it up to the Q&A. So please do post any questions you have for Geoff. And I'll warm up the floor by starting with a few of my own. So, key takeaways really today is understanding that the microbiome is at the center of our health. I mean, 70% of our immune system, you said, Geoff. And that current technology is kind of limited in accessing it. And actually, the biggest takeaway is this word ecosystem that you mentioned. I love that. I also found out today that the fiber that my mom would suggest to take is actually feeding my bacteria, not me, not myself. But what I want to hear more from you is on this word ecosystem. And we spoke about the gut microbiome today, but talk a little bit more on how that might communicate with your area of expertise in skincare, the skin microbiome, the gut skin axis, and how you see that in the future. So let's put our futures hat on in the way that consumers might behave in the future. Will they be choosing products depending on this technology that they use, whether it be ingestibles or any other product?
Okay, so ingestibles is obviously easy to deal with. We know that there are certain nutrients that we have that we ingest, whether it's superfoods, whether it's vitamins, all these things. We know there are things that you're ingesting which go to your skin and improve skin condition. And these are some which have got clear claims. Zinc is one of those. Vitamin C is one of those. There are vitamin E. There are things that are recognized by the European Food Standards Agency as claims that you can make on products that say these improve your skin. Niacin is in there, biotin is in there. I said there are all sorts of things. These do not get to your skin without going through your gut microbiome. They will metabolize them. And so anything like that, anything that gets into your system has gone through your gut at some stage. There are certain vitamins which are only made by your gut microbiome. And so you don't ingest them. These are manufactured in your gut by the bacteria that you have in there. And the bifidobacteria, the other major probiotic, produces a lot of the vitamins that are useful. So that's the one side, the nutritional side. On top of that, you have the immune system side. And so many of our skin conditions have an immune response side to them. So eczema has an immune response side of things. Acne has an immune response. Psoriasis, vitiligo, all of these are potentially immune response. They're to do with how your body interacts with the external environment. And redness, sensitivity isn't an immune response. It is, how your body interacts. And so if you can have a healthy immune system, you are less likely to have a problem with your skin. On top of that, there is that fact that the bacteria themselves are reaching the bacteria in your gut are putting metabolites into your system that go around your body in your bloodstream, and those metabolites will get to your skin, and the bacteria in your skin will respond to those because they are they're signaling molecules, bacterial signaling molecules, so they will respond directly. So your gut bacteria are talking to your skin bacteria, and that is a direct interaction.
Absolutely. And so do you think in the future, and possibly a good question is how far in the future, will consumers' behavior change then in the products that they purchase according to what this, say, monitoring device does? So say, someone takes in an ingestible, sees, what health kind of markers are missing, and purchase products according to that, whether it's for skincare or nutraceuticals or anything else.
So there are two sides to this. So the straightforward answer is yes, probably, but remember there is that causation correlation side of things. And so what these things, the first aim of any product like this, any diagnostic device, has got to be measuring whether an intervention you're having is actually having a benefit. So I'm changing my diet. Is it actually changing my gut microbiome? That's what I want. Change my diet to make my gut microbiome healthy. Is it having that effect? So yes, there is an aspect of working out what your gut microbiome is, and so taking something to improve it. But there is also probably the easier side is, I know I'm supposed to be having a healthy gut microbiome. I take my 5 a day. I take more. I reduce my red meat intake. I improve my fiber intake. Does it improve my gut microbiome? Is it having an effect? Is it having a benefit? Mm. And a lot of what the consumer wants is feedback to know that what they're doing is working. Mm. And this is one of the things that something like this can do for you, is to show whether what you're doing is having a benefit. Now, it doesn't necessarily mean that if you improve your gut microbiome, you're going to have better skin. But what it will show is what you're doing is having a benefit. At least that first step of what you're doing is having an effect on your gut microbiome. Therefore, it could be having an effect on your skin. And obviously, you can look in a mirror and say, "Oh yes, my wrinkles have gone down. Oh yes, I'm less red." You can see that. You can see what's happening. But you want that link to say why, what it is that you're doing, whether it's working.
I guess that answers, there's a question here, do probiotics work, or how about things like SIMprove? I guess it would help answer those type of questions, because so far it's a little bit of a trial and error, isn't it? When people, you try a probiotic and you kind of see if it works, or makes you feel
better. Do probiotics work? Yes. But what are you after? What are you hoping that they will achieve? Do probiotics in general end up, if you take a load of lactobacillus as your probiotic, does it inhabit your gut? Probably not. Unlike the skin, which is an arid desert, to be honest, there are lots of ecological niches on your skin, lots of bare space for something you apply to have an effect. Your gut is full. It is stacked full. There is no space for something you take orally to get in and have and start to grow. There are a couple of species which are in this probiotic lactobacillus that have been identified, which do have that, and they have been shown they're particularly sticky, and they're very, very good at sticking their elbows out and finding a space and so on. But just because they don't colonize does not mean they don't work. As they go through, they are metabolizing, they're producing the signaling molecules. The other bacteria that live there are responding to them. The lactobacillus produce bacteriocins, which are antimicrobial peptides. So as they're going through your system, they're producing these antimicrobials, which tend to kill the more pathogenic species. And they do this as they go through the system. And so these do have a benefit, even if they don't colonize. In fact, we know that some of them, you don't even need to have live bacteria to go through the system. You can have dead bacteria, and they will have the benefit as well, because as they go through the system, the bacteria in there see these bacteria. They don't know whether they're live or dead, but they interact. They see the surface of the other bacteria. They will respond to it, and so they will change their behavior accordingly.
That's very interesting. How would you, kind of addressing to future kind of participants in the hackathon, and given your experience, what advice would you give to our hackathon participants in the design of the capsule? Any kind of 101 applications that you're dying to see an ingestible capsule in? Or, we mentioned a lot of those signaling molecules. You mentioned bile acids, short chain fatty acids. Any specific biomarkers that are very important to kind of monitor?
So the thing to remember, if you're doing this as a consumer product, as opposed to a medical product, what you've got to do is you've got to have it give an answer which is relevant to the consumer, that you can say something about. And that's very, very important. It's not that you're taking it away and you're getting GPs and scientists poring over this. You need to keep it simple and be able to give a reason for it. So even something, just measuring the pH as you go through the system will be beneficial. It tells you it's something you can translate simply into gut health. bile acid metabolism, yes, it's there. The other metabolites, the key metabolites, like your butyrate, what is the butyrate concentration? What's your propionate concentration? Are you actually producing these, or do you have the bacteria to produce these? That's great. They're the key metabolites that get through to the rest of the system. But just straightforward things like temperature, like how crowded an area is all important. So that's what you can measure. Pretty much anything you can measure that you can say link back to the state of a gut or a gut metabolism. Remember, it's the gut metabolism you're really after is important. The other thing to remember as a consumer product, again, as opposed to a doctor's product, a medical product, is this is something that the consumer wants to be able to take and not only be cost effective, but not too icky, to be quite honest.
Going back to the user friendliness of the whole situation.
User friendliness. They want to take something that they can take, and it'll talk to their phone, and 12 hours later, oh look, I've got my readings on there. That's great. Having to send the readings away to somebody makes things complicated, because then that tells you what your gut was like, 3 weeks ago. And we know your microbiome changes according to your gut. If you had a curry last night, by today your gut is behaving differently. So you need to have something. You need something that is ideally immediate, is useful, and then you need to get rid of that ick factor. You don't want to have to collect something at the end of it and dig around in your poo sample, your stool sample, to take out that tablet that you can then wash off and send away to something. Ideally, you want something that can take the reading as it goes through your system and is disposable and can be flushed away. And obviously that then means it can actually be flushed away, and it is safe to be flushed away and go through the system. You don't want to be contaminating the sewer system with something as well.
Yes, you have to have, make sure that you're aiding individuals' health, but also not impacting the Earth's health is how I phrase it. Yeah. And given, what's your prediction? Given these kind of features, do you think it's a technology that will be mass adopted? Will it be a fad, or what's your prediction?
I think if you look at the current interest in standard gut microbiome testing, and there is a lot of interest in there, and you look at things like genetic testing, and there's a lot of interest in there, and both of those are limited over what they can tell you, and there is the ick factor with them, there is the time delay on them. If you consider the interest here, if somebody can provide something which gives the same or better feedback and advice to the consumer without all of those problems, without the time delay, without the ick, then the market is there and ready for it. People are looking for this sort of information.
Absolutely. Wow. Okay, guys, I've certainly learned a lot today. Please do share your thoughts. I have some great comments here. That was so interesting, Geoff. Or is it my brain biome thinking? Question mark. Super presentation, and hope to see many more in the future. So some great feedback. Please do share your thoughts as well in social media with our hashtag, so you can get a shout out. My takeaways today that microbiome is more important than our own genetics, and that we have several. We also have a brain microbiome, so you are thinking bacteria, and that what happens in the gut does not stay in the gut. So please consider that the gut is at the center of your health, and that it defines things like your immunity. So learn loads today. Please sign up for our next talk, which covers the translation of a PhD into ... foreshadowing there into a medical device. But join in. Thanks again, Geoff, for coming along and letting us know about the importance of microbiome and the future technology that awaits us.
See you guys soon. Have a lovely evening.
Bye-bye.